Hello there, and welcome to Smith and Sheridan, a podcast on the science and business of biotechnology. I'm Cormac Sheridan, and I'm joined this week by Salman Ho, who is the CEO of Medigene, a German biotechnology company which is focused on the development of T-cell receptor therapies for treating cancer. Salman, you're welcome to the show.
Well, thank you very much, Cormac. Delighted to be on the show and hopefully be able to offer a few insights this morning.
You're just about 2 years in post now with Medigene. You took up the job, you joined from Connect Biopharma. What did you see as the sort of opportunity with that company? What were the kind of technologies that it had that made you consider taking over the reins of the firm?
Thanks, Cormac. That's a really important question because what really attracted me to the company was the depth and experience of the science, as well as the commitment to innovation to develop the range of technologies that really inform the basis of what we would call our end-to-end platform. As I dug deeper and deeper into it as part of my due diligence, it became very, very clear that this was an ethos that had been developed over many, many years. So to give you a bit of background, if you recall, Medigene as a company had acquired a cell therapy spin-out from the Helmholtz University called Trianta, and was moving therefore into a pure play cell therapy company, which at the time in 2014 absolutely was their cutting edge technology. You know, we had started to see the very early data in CAR-Ts and starting to see TCR-T therapies, which were being talked about and initially being developed early in the preclinical stage at that point. And so that was really the attraction, particularly the ethos around doing the cellular therapies, because you're aware it was very, very hot as an area many, I would say 5-7 years ago.
At the same time, I mean, there was so much excitement around the development of CAR T-cell therapies in that decade. Chimeric antigen receptor T-cell therapies, for those who are not across all the jargon. And many people thought that the T-cell receptor cell therapies, or also the recombinant T-cell receptors, which I guess play a sort of an analogous role to antibodies, everybody thought that they would be the sort of the next big thing, that they would follow smoothly after the CAR-Ts and that they would greatly broaden the antigenic reach, if you like, of these therapies. Because obviously CAR-Ts are restricted to extracellular antigens, and it's really only a handful of antigens such as CD19 and B-cell maturation antigen have proven sort of to be viable or feasible for the CAR-T developers. And the great attraction of the T-cell receptor therapies was that you were essentially going to get access to a much, much wider domain of antigens, including fragments of intracellular proteins as well as extracellular proteins, albeit presented only in the context of major histocompatibility molecules. But that this was going to really broaden the reach and hopefully, therefore, allow developers to get really stuck in on solid tumors, which has been an area where the CAR-Ts have so far not really delivered. Has that played out, though?
I think that's a very fair assessment, what you've described there. I think the promise and almost the distinction between the CAR-Ts versus the potential for TCR-Ts has really been driven by this opportunity in solid tumors. So fundamentally agree. I would say as of today, that hasn't transpired, but that's not necessarily the end of the story. I fundamentally believe there are very clear indications that this is going to be the case, that the next round of T-cell based or T-cell receptor based therapies are literally on the horizon. And if we deconstruct some of the elements of why that hasn't happened to date and look forward to how they could change, there are a number of things. I think, first of all, there's the general market sentiment, which is around a little bit of hype. So CAR-Ts have initially not been super successful, and then all of a sudden, it's like buses, 6 have come along. We've seen greater and greater success on, I think, to be fair, a very limited number of extracellular targets, whether it be CD19, BCMA, CD20, CTLA4, etc. However, the success of that has then put certain expectations into the market of investors and I think potential pharma companies as to what the TCR-T therapies could do. And almost the expectation was, well, you now reached a mature stage, let's say, for CAR-Ts, where in your second, third, fourth, or even fifth generation CAR-Ts. Why can't you do this for TCR-T therapies? And I think if people just step back and say, well, look at the journey that CAR-Ts went through, it took some time. It took some time to iron out all the challenges of cellular therapies in general. But more fundamentally, I think you have now had an advancement in the science, which is absolutely critical over the past 10 years as well. What we thought was canon 10 years ago is now fundamentally different nowadays. I think a continuing thread through that time, though, has been the challenge of manufacturing cell therapies, which I think is one of the biggest issues that will remain the case. I do think that there are green shoots on the horizon that are very clear as well. I think we're seeing advancements in how we understand what a great drug product looks like now. For example, we know that the time it takes to manufacture plays a critical role in the expansion of the cells. And there are different philosophies. Let's be very clear. There are different philosophies, and ultimately it's only going to be proven in the clinic. But our philosophy, as an example, is start off with young, healthy cells. I'm not being ageist here at all, but young, healthy generally seems to be the thing here. And we start off in our entire process of generating our cells with young, healthy volunteers. But coming through to how we develop our cell therapy process development, we have a very short expansion period. So we only take 6 days. If you've looked at traditional products, that could be as long as 14 days or even longer. And what that does is that translates ultimately into a vein-to-vein time, which is the time it takes to take blood from a patient, go through the process of ex vivo manipulation engineering, expansion, manufacture, transport to and from the site, and then back into the patient. Historically, even with CAR T cells, that has been up to 6 weeks. Now, that period has come down over time as process improvement. We have guided to market that we are going to submit our IND within the third quarter of this year. We've been in negotiations and discussions with the regulators, both in the US and in Europe, to bring our expansion period to 6 days, which could mean a vein-to-vein time as little as less than 3 weeks. Now, certainly if I were a patient and you said to me, I got diagnosed on day 1, would you rather wait 6 weeks? Would you rather wait 3 weeks for the therapy? It's pretty clear.
Even though before you get to that, which is very impressive, definitely, and definitely sounds kind of like in the ballpark where it needs to be, but the choice, say, of antigen, how you actually design the product in the first place. There were some, what I find, the big sort of poster child for the dangers of T-cell receptor cross-reactivity was a therapy developed originally, and this is ancient history now, but this is Adaptimmune's work on a MAGE-A. No, it wasn't.
A3. Yeah, MAGE-A3.
Yes, you're right. And in a trial that elicited cardiogenic toxicity and 2 patients died. And subsequent work established that the TCR that they were using recognized an epitope on this cardiac protein called titin. But also then, later on, or no, around the same time actually, a trial led by the great pioneer of this work, Steve Rosenberg at the National Cancer Institute. And actually, I know that Laura Schendel, who's CSO, was a co-author on this paper, their account of a different preparation of MAGE-A3 targeted TCR-T cells in patients with either melanoma or esophageal cancer and sarcoma. They had profound neurologic toxicities in 3 of 9 patients, and they assumed not assumed, they established rather that this was a cross-reaction with a MAGE-A12 protein expressed in a subset of neurons that had never been sort of reported in science before. All that strikes me as being that antigen selection is, now I know this is 10 years ago and we have technologies around antigen scanning and epitope recognition that are much more powerful now. We have a much deeper lens into human biology than we had then. But it just struck me, though, that these trials did throw up profound safety questions, which means you really have to step very lightly in terms of your antigen selection process.
I think, again, your account of that is absolutely true and fair. And I would re-stress the point about the extension, expansion, and continuous understanding of the science. I think that's a critical part of what you mentioned. If you look at it now versus maybe 10 years ago, the antigen is still absolutely a key part of developing the eventual TCR-T therapies. But our understanding of TCR biology now is incredibly advanced compared to what we knew then. The standard canon from 10 years ago has been modified and modified and modified further. And what we know now is how do I best describe it? There are ways when you look at the way that a TCR binds to any antigen, okay? And the understanding of how that TCR interacts with the benefits of now much more powerful computing, we're able to look at protein engineering, structural biology in deep, deep depth on the one side, and can understand the impact maybe of a single amino acid chain in the binding domain as to the effect on the overall binding. In parallel to that, the issue around cross-reactivity is, do we understand whether or not the antigen that we're binding leads to on-target, off-tumor effects? So the absolute undesired, on-target, off-tumor. And because of the way that TCRs are developed in the human body, we really need to understand, taking a step back on some of the basic immunology, that the repertoire of T-cell receptors that we would develop in a normal human being is actually a repertoire of very, very weak TCRs to begin with. They've gone through something called central tolerance. They've had positive and negative selection. And what you have in the periphery now are very weak TCRs. And that's by design, because you don't want autoimmunity and self-recognition. The other concept, though, that is very clear now is that these TCRs if you look at the binding sites for a TCR, and I won't drop into a huge level of immunology detail, but you've got a very limited number of amino acids that play a role in your binding site versus binding to a peptide. It may only be 8 to 13 amino acids that play an important role. But even if you think about 8 to 13 amino acids, one variation may lead to an increase in binding, one variation may lead to a decrease in binding. But these TCRs, because you have a very limited number, have to recognize close to billions of potential peptides, both self and foreign, viral, etc. So there's a lot of polymorphism. And so recognizing that a peptide may still bind to a TCR, even if you had 8 of exactly the same, or 7 of exactly the same, or 6 of exactly the same of the amino acids, leads to this cross-reactivity. Because you can get a TCR that binds fantastically well to the desired peptide, whether it be MAGE-A3. But because, let's say, chitin, for example, shares a similar 7 or 8 or 6 amino acids to the MAGE-A3, you get this cross-reactivity. And so this is the parallel bit that I was talking about now, where the screening for this is vastly, vastly improved. There are a number of in silico tools, machine learning and AI now that run hundreds of thousands, millions, tens of millions of potential epitopes, changing one amino acid at a time to predict binding, but also now to screen for potential other known sequence peptides of the body. that just gives you an idea of the mismatch. I think the final part, though, on the safety is there are other screens now where, I know companies that screen, for example, 100 different allo HLAs. They look across 20 or 30 different healthy cell lines to pick up any evidence of cross-reactivity and potential safety issues. And that's now become de facto standard in our industry because of these issues in the past.
Yeah. And what's also fascinating is that, with that NCI trial, especially, that there was such a variation. It wasn't that all patients had this neurotoxicity, because there are obviously biological variations in expression of any gene. And I think that's interesting and complicated. But in terms of how the industry, though, is taking forward these particular therapies and sort of the antigens that they are selecting, it is reminiscent to me of the CAR T story, that we still only have about a half a dozen or so antigens that are in play. I mean, say your IND is for your lead product, MDG015, that's going to target the NY-ESO1 antigen, which, say, Adaptive Immune is already in a pivotal trial with its TCR cell therapy, lete-cel. I'm not going to pronounce the full stepping gingerly here. So, and I mean, say certain of the MAGE antigens are still in play. I know as well, your pipeline, you're doing an awful lot of the KRAS mutations. So there's a kind of a convergence, isn't there, on terms of the antigens that people are targeting?
I think that's, again, a very good assessment of where we are. And I'd say, though, that as a clinician, fundamentally, we validate the success or the potential success of a target, not from the preclinical work, but by the clinical outcomes that we see. So at the point where we have brought many more targets into the clinic and demonstrated clinical efficacy, that is either the same or potentially better than the standard of care, that is the point where we often say that is a validated target. So we have scientific validation and we have obviously clinical validation. By default, because we are 10 or more years behind on the CAR Ts, the number of so-called clinically validated targets is much, much less. And I think the key point on your word convergent, which I think is a good term, really has to understand the context of pharmaceutical development, biotech development. Every company wants to have the highest possible chance of success. So in general, we attempt to de-risk in many ways our development processes and our development plans, whether it be at a strategic or even at operational level. So starting off with scientifically validated targets really puts that first step in place. Okay, what is the scientific validation for these targets? And as you said, there may be 10 or so targets that have had sufficient scientific validation to date that allows us to say, right, this would be an interesting target to take into clinic. NY-ESO is by far the largest one. MAGE, PRAME, KRAS, TP53, Wilms tumor 1, etc. I could go on. There are, I say, 10 to a dozen of these. Until we get the clinical validation, nobody's going to stand up and say there is success, and therefore people then rush into it. And I'd say the final point on this is actually more broadly at the pharmaceutical industry. What is hot now from a hype perspective would be something like a GLP-1. Incremental variations on that have driven a huge amount of investment. So having a single target per se is not a bad thing, but the question is whether ultimately the efficacy, safety, convenience, cost improves time after time after time.
And then also, I mean, obviously, some of these targets that are currently being addressed with TCR-T cell therapies can also be addressed with recombinant or soluble TCRs themselves, just using the protein part as sort of a drug. And obviously, the KRAS story from a couple of years back, the small molecules have certainly started to prise open the area of KRAS mutations in cancer. What do you think are the sort of advantages of taking the cell therapy route rather than the soluble TCR route in certain instances, or the small molecule drug route in others, to interrogate these targets in the context of various cancers?
And look, I think the overarching premise and almost the number one advantage of any potential cell therapy is that it could be a one and done. It could offer a functional cure over, let's say, repeated small molecule ingestion or administration on a daily, weekly, monthly basis of an antibody. So I think that's always been the hope for a cellular therapy, provided, and this is, there's caveats, it's provided it demonstrates the level of efficacy required, but also it has the persistence in the body, because these are living therapies. So that's the fundamental thing. But along with that, particularly for TCR therapies versus in general cellular therapies like a CAR-T, you also have advantages or additional elements such as the PHLA specificity. That's an absolutely critical part of the value proposition. And it's different sides of the same coin here, where you get this great potential specificity for using a TCR and using the PHLA interaction as the basis of that. By definition, you will have a slightly smaller population because you're HLA dependent now. But there are always ways to get around these things. So there are technological ways to deal with persistence. So for example, we obviously focus a lot on our co-stimulatory proteins. We focus a lot on bringing in the best possible drug product that demonstrates the highest level of T cell subsets that have central memory or stem cell like memory. We've demonstrated and discussed in public domain that we can get a TSCM, TCM fraction to be around about 80% of the cells. So we fundamentally believe that will have a major benefit in terms of persistence in that. But coming back to other technological, if you have one HLA and therefore a limited population, by definition, people are now expanding and looking into this concept of building a warehouse or library of potential therapies. So it will be the same antigen, but now different HLAs. One final point I was just going to quickly add was...
And by the way, too, would each of those need to go through its own development process so that you would need to maybe do, say, even potentially 6 or so different actual therapies to cover a broad patient population?
So as an individual therapy, yes, but the kind of companies that have started this process earlier than ourselves have had some very positive interactions with the regulatory authorities. So whereas before, particularly if you go back to the examples and the history of CAR-Ts, you would have to submit a new IND or investigational new drug application every single time. I think FDA is now much more comfortable in allowing you to use an existing IND, but to introduce new drug products with potentially different HLAs. Almost the next step to that was the other piece that I was going on to, which is we know that tumors are very heterogeneous in terms of their expression of antigens. So, there's plenty of data out there that shows that for a given tumor, take gastric cancer, you will have expression of NY-ESO, of MAGE, of PRAME in there. So if you wanted to have the best possible chance with the largest population, not only do you have to have one target of multiple HLAs, you need to have multiple targets with the same HLA. That step in the regulatory process is ongoing. The single target multiple HLAs has been addressed by FDA. They've given us a regulatory pathway, or given the industry a regulatory pathway to doing that. I say the next step will be multiple different antigens, same HLA.
Yeah, nothing as yet has reached IND approval stage with that type of an approach.
No, only for the multiple HLAs for a single target. Yeah.
And then your own upcoming trial, you're on track for an IND submission in the third quarter this year, is that right?
Yes, we're delighted. We've been really going great guns to make sure that is an operational reality, and we remain absolutely on track for that.
And have you selected the clinical trial sites?
Absolutely. So we announced earlier this year both the selection of our contract development manufacturing organization, the CDMO, that's AGC. We've also selected our CRO. We haven't disclosed who that is, but we've been deep in feasibility now for many, many months with clinical trial sites in the US as well as in Europe. So we've, just as an aside, we've also committed to submitting a clinical trial application in Europe in the fourth quarter of this year.
So first patients could be dosed early 2025, presumably, all going well?
Let's say with a fair wind behind us, we aim, if we can do a further round of financing, to actually start the trial subject to IND approval this year. First patient enrolled this year. That would be our case with the financing in place.
Okay. And I know, obviously, as we were saying earlier, there were other players in this space as well, directly targeting the same antigen. Now, that's not to say that there wasn't plenty of room, and especially for any novel technologies that might enhance the activity. But how do you differentiate yourself from better funded and competitors who are further advanced down the clinical development pathway?
I think the first thing to say to that is we recognize that there are companies who are further in the process towards a marketing authorization. That's very clear, and that's a fact. However, I think the history of our industry has demonstrated those who come first are not necessarily the best. I think there's multiple, if not, that is almost the rule rather than the exception. I think that is where the science has played a big role. So the way that we differentiate ourselves has been built on obviously understanding our own capabilities and the evolving science, but also learning from what our competitors have done and their data either in preclinical or now even in clinical. So we make a very clear play for what we believe is going to deal and deliver the ideal TCR-T therapy plan. First of all, you have to start off with the best possible T-cell receptor. We have processes that we believe are very unique to us. There's obviously a high degree of overlap as well with the techniques that other companies use. But in our secret sauce is a lot of the way that our algorithms are written, our experience of multiple decades looking at T-cell receptor development, and just our very strict standards for evaluation and also thresholds. I am in no doubt that some of the TCRs that we reject would be selected by other companies because we take very high standards. So that's the first key thing. The second key thing is you've already touched on. How do you protect a T-cell receptor T-cell in a solid tumor? Because we've seen with PD-1 inhibitors, with other immuno-oncology products, that the tumors destroy the T-cells, whether they be human T-cells that have been unengineered or even engineered T-cells that are being administered as part of clinical trials. We firmly believe that you need to provide some mechanism that allows the T-cells to, first of all, survive, but then actually thrive within the tumor microenvironment. And our methodology there is to introduce what we call co-stimulatory proteins onto the T-cell to support the activity that you get from binding the T-cell receptor and then causing that tumor cell killing. And we use PD-1/4-1BB as our first co-stimulatory protein. So PD-1 is a well-known mechanism of action. 4-1BB is the intracellular part of the activation pathway, and that leads to much greater persistence. And we've demonstrated that in our preclinical work. We can show that with a single administration, without providing any further media to support the T cells, in vitro, we can get up to 6 or 7 cycles of repeated cell killing from a single administration with further tumor re-challenges.
And is there any concern, though, that if you have those kind of turbocharged for this, I mean, that's just a metaphor, but if you have these highly active T cells, that if you have excess activity, that you might run the risk of having these on-target off-tumor effects?
I think there's always a concern, and the way that we mitigate that is the effect of the PD-1/4-1BB is what we call gated. It's absolutely gated only by binding the TCR to the target. So that's why you see the interplay now, the critical need for a highly specific, highly sensitive and safe TCR. By allowing you to do that, you can then add in a co-stimulatory protein. The final part of that little element is also to think about the dosing of cells, because one part of traditional, almost the pharmacology and the pharmacokinetics, what is the level of cells that you would need to generate your minimum response versus your maximum tolerated dose versus your dose limiting toxicity. Now, with our process, as we mentioned, it's a very short process, 6 days for our expansion, where we've been able to demonstrate the levels of efficacy we've seen with such a short process in vitro. So we will be potentially starting at dose levels significantly lower, part of our dose ranging and dose expansion trial, than we see with our competitors. As an example, we see billions or tens of billions of cells being used now by our competitors. We will be starting in the millions.
Wow, that's very interesting, actually. Very interesting.
Yeah. And that was really the final part, is the drug product itself plays such a critical role. Having young, healthy cells through a short process with very high TSCM, TCM levels, using only a CD8 fraction, to have almost a pure CD8 fraction as the basis of our T cells. This kind of level of ethos and the thinking, learned through other people's mistakes, but also our own experiments, that we feel could give you this differentiation.
And if you hopefully put everything in place, you have the financing in place and everything is up and running, that if your first patients are being dosed before the end of 2024, your first data may come, late 2025 maybe?
Exactly that. And to be clear, I think what the benefits of a great TCR with an armoring and enhancement and with a fantastic drug product of young, healthy cells with a pure CD8 fraction and high persistence is that not only do we expect, or at least hope, that the efficacy that you would see, and here you'd say something like an overall response rate as the marker, that we would hope would be at least as good, if not better, than what we see. But fundamentally with the CSP, we would also expect progression-free survival and duration of response to be extended well beyond what we see with current therapies that are unarmored and do not have this high TSCM, TCM fraction. That's really where we expect to win.
Yeah. Okay. And obviously, as is always the case with biotech, time will tell. But I know too that you have partnerships with BioNTech and Regeneron as well. I'm not quite sure what the status of those are.
So with the first one with BioNTech, I think we announced very recently actually that we have extended the partnership with BioNTech beyond the initial 3-year term that was announced back in February 22. So it's going now beyond February 25, and we're working currently on a number of targets for them. Obviously, that's confidential what they are, but the scientific partnership has been absolutely top-notch. Very happy with the outputs that we've provided. We've really shown the capabilities of what Medigene can do in delivering potential best-in-class T-cell receptors. With Regeneron, we actually announced back in 2022 that the research and collaboration agreement had been stopped, but we have an existing asset that they have actually progressed. So this is a TCR that we licensed them for MAGE-A4. The original strategy from 2seventy Bio, which then sold the asset and all their cell therapy assets to Regeneron, was initially to develop that in the US first. They then pivoted slightly and developed a strategic partnership with a Chinese company called JW Therapeutics, with I understand the belief to be that they could generate data in China through an investigator-initiated trial process quicker. I think that's been borne out in many ways because JW Therapeutics and 2seventy Bio at the time announced the start of a phase 1/IIT trial in China at the beginning of this year. We hope that that also becomes part of the clinical validation, as would be the original PRAME A2 that we licensed to BioNTech. As soon as they get into IND, into clinic as well, that will help further validate our approach to generating these best-in-class TCRs.
Okay. So it's been a sort of a long haul, but you were sort of obviously entering a new phase in the company's evolution.
Absolutely. Look, the one thing I think that is demanded now of CEOs, of management teams, is resilience. I can definitely say that we have a very resilient team. The science really drives us on. We look at the quality of the science. We believe in the translatability of that. Our role in the kind of less scientific functions is to convince potential investors and partners that this science really will be translated, not can be, will be translated. And, at the moment, I think with the markets, we may have opportunities as the sentiment improves where people are prepared to take a little bit more risk. Rather than having absolute certainty, they may go on, this is such clear scientific evidence. We would want to continue to look at this very, very actively.
Okay, I like that idea that resilient companies develop resilient T-cell therapies.
I may have to steal that one, Cormac, actually. So you better copyright it quickly.
But on that note, thank you very much, Selwyn, and best of luck with all that happens next.
Thank you very much, and thank you again for the opportunity to discuss some thoughts, and look forward to the future as well.
Thanks for listening, everybody.